this stage we'll hand over to Leslie, who'll be chairing the plenary session.
Hi, so I'm Leslie Jones from Cardiff University, and I'm co-chairing this session with Davina Hensman-Moss, and Davina is going to start the session off for us.
Hello everyone and welcome to this session. It's lovely to have you all here, and it's my great honour to, with Leslie Jones, introduce you to this session on genetic modifiers of Huntington's disease, DNA damage and somatic expansion of repeats, where we have three fantastic leaders in their field talking to us today. For those of you who are less familiar with the background, this is an area that's demonstrably important in Huntington's disease. There have been some large-scale genetic studies that show that the things that we'll be hearing about make a difference in people. And I'm really looking forward to learning more about the underlying mechanisms acting at the level of the DNA and proteins that interact with it, and how that might change outcomes for people with Huntington's disease. So the first speaker we have today is Professor Vincent Dion. He's a professor of DNA repair and neurodegeneration at the UK Dementia Research Institute at Cardiff University. He's a rising star in the field, and it's a pleasure to have him here. He has come from an assistant professorship in Lausanne, postdoctoral work in Basel and a PhD in Houston, and in 2019 gained an Academy of Medical Sciences professorship. And Professor Dion's going to be talking to us today about gene editing for expanded CAG and CTG repeat disorders. So over to you. Thank you.
Thank you very much for the introduction and for having me here. I think you should be able to see now my screen. And so I'll jump right in. What I want to try to talk about today is gene editing to try to contract repeats that cause Huntington's, among other diseases. The idea is that longer repeats cause more severe disease at an earlier age. And so if you can take a long repeat and shrink it down to a non-pathogenic size, then you would expect some benefit to patients. Conceptually, that sounds really easy, but in fact, it's actually quite hard to achieve. What people have been doing in terms of gene editing has been taking this concept and saying, okay, let's remove the repeat track altogether. And so this has been done in multiple different systems for multiple different diseases. And the general concept, although I apologize for summarizing the work of a lot of people in half a slide, the general concept is to try to induce double-strand breaks using CRISPR-Cas9 on either side of the repeat track and excise the repeat region, including some flanking sequences. This is error-prone repair that you're hoping to achieve that would remove the underlying cause of the disease. And so the good news is that this seems to work. So in cases where it's been looked at, a lot of the molecular phenotypes are being corrected by doing this. There's some issues with it, in particular rearrangements that tend to occur locally when you have one break, or you can flip them around as well. The guide RNAs, you need two guide RNAs with potential off targets. You're not necessarily allele selective. You might, unless there's the SNPs that you're targeting that is present in the patient. And so with these caveats in mind, we actually tried a different approach where we used a Cas9 nickase that cuts only one of the two strands of the DNA, and we targeted specifically the repeat track. And what we find is that, as I'll show you, is that we get contractions out of that. That was completely unexpected, but this is what you get in science. So when we first started doing this, we used a reporter line, so a GFP reporter line with 2 exons of the GFP in green and a CAG repeat inside the intron here. And if you get an expansion, you end up interfering the splicing of this reporter, and your cells are not very green at all. But if you have a contraction, then you restore the splicing, and then you end up with green cells. So GFP plus cells is what I'll be referring them to. And so in this line, we can, the cell line, so this is just hex cells. It's not a disease model. It's there for screening for things that may be interesting. So what we did was to transfect in our Cas9 as a control. So I'm just showing you sort of a model data to give you a sense of what the quantification is like. So we transfected in our Cas9 without a guide. We look at GFP expression with flow cytometry, and then we establish two gates, a GFP minus gate and a GFP plus gate. And so if you sort cells from these two gates, you get expansions in the GFP minus and contractions in the GFP plus. And so this is what it looks like. So you normalize it to this, and that's the dashed line in the background. And then you do the experiment with your guide RNA targeting the repeat track, and you look for how much of your new experimental distribution is in one of those gates. And so you can do this multiple times, get error bars and the like. And so, oops, I'm going to start here instead. So so when we transfect in the Cas9 nickase, that cuts only one strand of the DNA, and then we can do this on one or the other. It works both ways. We end up with a bias towards GFP plus cells. And so that's suggesting that we get contractions. And in fact, we can follow this up with a classical small pool PCR to look for repeat size distribution in these cells. And the way it works is that you take your population of cells, you isolate your DNA, and then you dilute it down to just a few genome per tubes. And this is required to get around the bias against amplifying long repeats. But because you have so few genomes, you have so little product, and you have to run a gel, transfer the membrane, and do a southern blot. And this is what it looks like. This is a cell line that had 100 repeats. We're transfecting in the Cas9 nickase without a guide for over 12 days, 3 different transfections. And so you see variability. So this is the instability of the repeat track. It's expected. But when you have the guide, you end up having contractions down to non-pathogenic levels. So this was actually quite exciting. We could move it to a different cell system, specifically in this case, lymphoblastoid cell lines from myotonic dystrophy individuals, which have an expansion of a CTG repeat at the DMPK locus. And here we transduce the lymphoblastoid lines with a Cas9 nickase, lentivirus, and then with a guide RNA lentivirus, and then we looked at time points. This particular individual had 1,600 repeats on one allele and 5 on the other. And what you can see is within 5 days, we had lost the very large allele. And by a month in culture, we had very few expansions left. So this is quite striking. It doesn't always happen like this. In the lymphoblastoids, they seem to be particularly efficient at doing this. Right, so what that meant is that it wasn't locus specific. The next question we wanted to ask was whether it had to do with repeat sizes, like, well, so we could get specific clones of our reporter line with different repeat sizes, but that are otherwise isogenic, and redo the experiment. So everything I've shown you was with the 101 repeat here. You can see that 42 repeats, not much happens, but at a longer repeat track, you get also your contractions. So I'll say right off the bat is you're looking at this and you're thinking, well, Huntington's is at 42 repeats is long enough to cause the disease. So would that even be useful? So if you think about a two-step hypothesis where the first step is repeat size, is the repeat size you inherit, and then the second step is somatic expansion, we would be targeting the second step in this scenario. Okay, so then there's the question of the safety, which I'm going to spend most of the rest of the talk on. And that has to do with off targets, for example, and rearrangements locally. So for off targets, what we did was to sort cells. So we sorted 9 different clones, single cell isolates that would grow up, and that have a contraction due to the nickase. And so we know that the nickase was active. And then we sequenced 7 endogenous loci with CAG repeats in them. So we're looking at at least 126 alleles, and we found no changes in any of them. So repeat composition is the same, repeat size is the same, and flanking sequences to the repeats were unchanged. So that suggests that the off targets are lower than our cutoff for the sensitivity of this assay. In terms of mechanisms, I'm not going to go too much into this today. What I just want to point out is that we think what's going on is for an expanded allele, we end up with multiple nicks on the same side, on the same strand that can create a gap. These DNA gaps can fold into secondary structures, and then you can ligate on one side, leading to a heteroduplex that can be processed further after to a contraction. But one of the mechanistic details that turned out to be quite interesting and quite useful is the fact that we can actually ask whether transcription is required for those contractions. And so here what we have is we have an inducible promoter on this reporter gene that's driven by doxycycline. So what you can do is we can transfect our Cas9 and our guide with or without having doxycycline on there. Wait for the Cas9 to degrade, and then turn on the transcription to read the fluorescence after this. And this is what the result is. In fact, all the action is at the very end here, where you have everything together. You have the transcription is on, you have the Cas9 nickase, and you have the guide against a repeat track. And so essentially 100% of the contractions are dependent on transcription. So what that meant was that we could actually make a stable line that had a single copy integrant of our reporter with an expanded repeat in it. We had a guide RNA construct that was integrated and a Cas9 nickase that was integrated. And then in theory, you should be able to turn on the transcription and watch the repeat track shrink. And this is what happened. You can see here we have 3 lines. The first one on the left doesn't have a guide, so transcription on and off using a bulk PCR assay, you don't see much differences. If you use 12 repeats with a guide, then you see there's no much movement going on at all, and so that's because the repeat size is too short. And then the third one here is where you have the transcription is on over here, and you can see within a few weeks, you get a shrinkage of the expanded repeat with accumulation of this of a short band here. So at this point, the next question we wanted to ask was whether there were large-scale rearrangements like we had seen with double-strand break-inducing approaches for gene editing around the repeat track. And so this is coming from the fact that in yeast in particular, if you use Cas9 to induce a double-strand break within a repeat track, you get massive rearrangements and deletions. And this is the work from Guy-Franck Richard's lab. And so I think this is a particular concern. And so we opened a parenthesis here and went into bioinformatics to try to sequence this locus. Anybody who's tried to do this will know that it's exceedingly difficult for the bioinformatics in particular and the sequencing for expanded repeats. So we ended up developing an algorithm to count repeats called Repeat Detector. So we have, and I'm happy to tell you afterwards how it works. In essence, for the aficionados out there, you get a circular profile, and then you align your reads to that, and then you look at the highest scoring repeat size for your read. So this worked really well. It worked. We tried it on smart sequencing platforms and on MySeq sequencing, so that specifically with a cohort at the bottom right here of HD samples from Leslie Jones and Darren Monkton. Those, it turned out to be just as good as Scale HD from the Monkton lab, but portable to different loci as well. So we were pretty happy with that. We took that and then we inserted into structural variant calling pipeline. And that's because those structural variant calling pipelines are notoriously bad with expanded repeats. they lead to positive, to false positives, because you have changes in repeat sizes that throw them off essentially. And so what we did is to insert repeat detector in the middle of this flow, this workflow, this pipeline. That way we could essentially control for repeat size. And this worked really well. We can now detect rearrangements when they're there. We are working on this, so I'm happy to talk more at some other time on this. But then we had a tool to actually answer whether we had large-scale rearrangements at the repeat tracks upon nickase-induced contractions. And so we took those same samples that I just showed you and did PacBio sequencing on them. And this is what you get. So you have count on one axis and repeat size on the other. And so in blue is before the experiment. In green is what happens if you have transcription that's off. So they're largely overlapping. And in red is our contractions that I've shown you. So this is 6 repeats right there. That seems to accumulate over time. And so we ran the pipeline and we found 0 changes other than repeat sizes. So, and I can say for having run other samples that this was unusual. And so we were quite pleased with this. And so the conclusion from that is that we don't see what people have seen for double-strand breaks in yeast. We don't see these large-scale rearrangements in this case. And so I think I'm a bit early, but just so maybe there's time for questions. But there's a couple of outstanding questions that we're addressing at the moment. One is whether it occurs in non-dividing cells. That would be a critical question. But I was hoping to be able to tell you this today, but I cannot. It's going to have to be a couple more weeks, I think. And then there's the frequency of target mutations. I've told you that it was less than about 1%, but how far below that 1% mark is difficult to say at the moment. Specifically, you want it as low as you can possibly get it. And so 1% would probably be not very good for a translation into a clinic, but much lower than that would be better. And so I think that's really important to find out. The other thing is the molecular mechanisms of contraction. In that front, we've made some progress. There's a poster from Laura Herati in my lab, a PhD student. She's doing some really fun work on trying to see whether how FAN1 might be involved. So this is an endonuclease that is a modifier of somatic expansions, and it turns out that it seems to work in preventing the Cas9-induced contractions, which was surprising. So her poster is C03. And so with that, I just want to quickly thank the people who have done the work. So Lauren, Cinzia, and Melvin have done the work that I've shown you on the GFP assay. All the bioinformatics was done by Ali and Eleanor. We have a number of collaborators, and I forgot to update the slides, so I'm going to make a shout out to Darren and to Leslie Jones, who have provided data before publication for us to play with our repeat detector pipeline. And so with that, I'm happy to take questions or let other people talk. Thank you.
Thank you very much for that great talk, Vincent. We do have, I think, a couple of questions. Can nanopore sequencing solve the problem of precisely determining the length of repeat genes, such as the Huntington's genes? From Veronica Maria.
Yeah, this is a loaded question. So I'll give you my opinion. So I think there's a possibility of getting like an average repeat size for a sample, right? So, but a distribution of individual reads, that's going to be a problem. We've tried our repeat detector pipeline on MySeq data that worked very well. We tried it on PacBio data that worked really well. On nanopore data, we only had access to some C9orf repeats. And there were so many mutations within the repeat track that often we couldn't tell where the repeat track started and where it ended. So So what we got from this is that there might be, we don't know for CAG repeat specifically, and we haven't tested it, but it's likely that the error rates are much higher in nanopore, and that may be difficult for that reason.
Yes. So how predictable are those Cas9 nickase contractions, and how predictable are they across different cell types, do you think?
So we don't know this. We've not quantified this specifically. It's not trivial, because then you would have different levels of the Cas9 and the guide at the same time as changing the cell type. So you have multiple variables at the same time. Anecdotally, the lymphoblastoids for the DM1 were about the same. We're going down to about 5 repeats, right? So 6 repeats is what we find in the reporter cell lines. So, but this is just, 2 cell lines and with lots of variables. So I wouldn't want to bet too much on that.
And one more question, if you could be fairly brief, please. So from Anne Rosser, if it turns out that contractions don't occur in non-dividing cells, would this limit applications to use in pre-implantation manipulations? Right. So it depends. So is the short answer. The longer answer is that you might have a situation where you have a contraction on one of the strands and not on the other, in which case you would still transcribe a shorter repeat. And so that would be something that we would need to look at. So it depends a bit on the exact mechanism, is the answer. Okay, so I think we have to stop there, and I'm going to hand over to Davina at this point. Thank you.